So I think we'll begin. Welcome, everybody. I believe that this is our first SILT webinar, and so I'm pretty honored to be your host this evening. My name is Sarah Ashpole, and I am a research scientist who does biodiversity research in the South Okanagan, and I'm currently at home just across the border, about an hour south of Ottawa. So 10 o'clock my time, so a little bit late. And we actually have some of our guests this evening that are coming in from all over the place. So a lot of range of individuals. Our presentation tonight will probably stick within about an hour. I'm going to give a little bit of a background, and then we have our guest speaker, Jason. In many ways, Jason and I discussed this about having a little bit more informal setting. And so there's an opportunity for you to ask questions. We have a Q&A, and we also have a raise hand and a chat. You are actually all muted, but I can unmute you if I make sure I can use the technology. So I'm just going to share my screen and then tell you a little bit about SILT. So this is, as I say, our very first webinar. And I have been now with the Southern Interior Land Trust for about 2.5 years. And this is a land trust that formed in 1988, and it is a registered non-for-profit charitable organization. And our goal very much is to conserve and restore wildland for habitat for wildlife. And in many ways, this is spaces that humans very much enjoy as well. So a lot of our properties are part of, the human matrix, and we really do enjoy people getting out there and being educated on our land. And we have about 530 acres of land. And I'll have a few moments to tell you about how important it is, the funds that we have received, particularly for Site 16, which is our talk this evening. But we do accept donations in many different ways. And we have a wonderful new program on our website where you can be a land steward for a pixel of land. And there's a quick little button on our website where you can donate. There's also ways of donating through gifting of land and through bequests. And we are a registered charity, so we can provide income tax receipts as well. All of our board of directors are volunteers and donate their time. And we have actually quite an eclectic group of individuals. And I would really recommend any of you who are interested in being a part of a land trust and a board. We are always looking for board members, and we do definitely have some skill sets where we're focusing in on, but you will see that we actually have individuals from a really wide range. And we do actually have an executive director, and our executive director is Al Pete. And Al Pete is probably the face of SILT that you see the most often. And I'll also point out to you our president, Judy Steves, who's been a cornerstone of the organization. So what are we going to talk about tonight? So tonight we're going to talk about Lot 16, and I always have a problem pronouncing the name. I always want to say Bourguiba Spring, and this was acquired in 2022, and it is a 41-acre lot. And this place here, if you look on a map, these are our silt properties. We are right down here by Anarchist Mountain and Asuyas. And this is a very steep grassland area. It has a beautiful ravine in it, has groundwater spring, which is really important and critical for Haynes Creek. And it's one of the big things that drew us to this particular attention of this dry grassland system and this permanent creek bed, because it's really ecologically important at a provincial level. And we could not have done this without an extraordinary amount of participants. And so purchasing of this land did be very much a grassroots level. It incorporated funds from the South Okanagan Conservation Fund, the Wild Sheep Society of British Columbia, the BC Conservation Foundation, the Okanagan Smoking Park Society, the BC Park Foundation, the Government of Canada's Natural Heritage Conservation Program, part of Canada Trust Nature Fund. And there were, in fact, dozens of nonprofits that donated. And we even have here tonight some of the individuals and land stewards who actually helped donate funds. And I think the reason why it's so important for me to tell you of all these different individuals is that we play really well in the sand. We actually really do like collaborations, and we have a really strong history of collaborations in conservation lands. Um, if you look at this particular site, it's highly biodiverse, and that's going to be one of the reasons why we really wanted Jason to join us tonight, was to highlight the biodiversity. When we look at properties, we do a really intense assessment of their ecological value. And some of these are really tiny sites and some are much larger sites. And they often have on them unique habitat properties and unique species. Alpete does a lot of assessment to make sure that every dollar that's invested has great ecological value. And as a whole, kind of helps with connectivity. And so some of my most favorite species on this property are like the rattlesnake, large number of rare species like the badger. This is a badger den that you can see on your far right. So a really beautiful property. And this is as if you're standing on top of it looking down at Osoyoos. So it's high, um, beautiful views. It's actually quite extraordinary. So really, a special little place to be. And it's actually accessible. And so it's one of those sites that's not particularly easy to get into because there's not a parking lot or anything, but it's right there at the highway. And I've had the pleasure of going up the creek bed with Jason and with Al Pete. And Jason has also brought out other board of director members up that area. But you better have your hiking shoes on because it's pretty steep. So before I introduce Jason, I want to let you know that our our dollars very much are put to every single penny to use, and you can donate many different ways. And so I just wanted to point that this is our website, SILT trust.ca, and there's a button at the very bottom there that you can push on to do a very quick donation. And your funds go into our daily operations as well. And many of those things are, they're hard to put tangible numbers on them. So we might be having the purchase of land through large donations or through grants, but we actually do a lot of maintenance and manage. So these properties are visited on a regular basis. We have landowner stewardship, connections to maintain relationships with people. We have management and restoration actions. Some of them are at small levels, some of them are at very large levels. And so the operation of our funds day to day, week to week, month to month, actually come from individual donations like yourselves. And so I just want to say thank you to everybody who's donated and to encourage donation. And no donation is too small. No donation is too small. Okay, so I think we'll move on to our biodiversity research. And one of the things I want to highlight is as an organization, SILT doesn't pay researchers to do work, and we rarely actually have the opportunity for researchers to do work. And so we were very excited when Jason expressed interest in doing research on our site and was actually willing to do this in complete free of charge, like completely the resources of EcoLogic, the materials, the equipment, and his time. As you will see, it's pretty extensive. And so I think it is a nice thing to share with you that we do have research happening on our sites, and Jason is a great example. So if any point during Jason's presentation you would like to ask a question, I will try to moderate in real time. And so you can put a question in the Q&A and I will interrupt Jason at his request to clarify or to expand on an idea. But there will actually be also a moment where there's a break between projects where we can open up also to questions. And then you also have the opportunity in the chat, which is public, or you can make private, to give feedback or little chat amongst yourselves or in support of what Jason's doing. So there's a couple different ways that we can do this. And so I think what I'll do is I will stop sharing Jason, and we don't have any questions as of yet. So I'm just hoping everybody can hear it okay. And I will turn it over to you.
Great. Let's see. How'd that do?
Yeah, perfect.
Okay. Well, thank you very much, Sarah, for that lovely introduction, and thanks to the board for being supportive of my flights of fancy with relate as it relates to what passes for my free time these days, which most of the time I'm spending in the woods or elsewhere, and so what better things to do in my spare time than also play in the woods? I responded probably back in 2022, maybe early 2023, to a notice that Southern Ontario Land Trust had put out for a call for property stewards. And having recently relocated back to the Okanagan, where I grew up, it seemed like a real nice opportunity to get involved with a local conservation organization. Just a little background on myself. As I mentioned, I grew up in the Okanagan, in Kelowna. Left left home in the late 80s to pursue my education, and which eventually led me through my PhD on cerulean warblers in the Eastern Canada and South America. Ended up being a college professor post PhD for about 10 years, and then I've been in the private sector for the last sort of 15. My background is a wildlife biologist, but a more broadly trained terrestrial ecologist. Um, I currently own and operate two consulting companies, one of which is EcoLogic Consultants that provided, as Sarah mentioned, provided some of the materials for the work, and then Campfire Heritage Services, which is an archaeological services company. And it really has been my pleasure to get to know the SILT team, to get to know the broader naturalist and conservation community in the South Okanagan once again, and working with SILT on a few different research projects with other collaborators at Bourguiba Springs has really been uh, exciting for me. As you can see from the title of the talk, I didn't come up with something super pithy for the presentation, but what I wanted to talk about today is how the notion, which for those of you who are ecologists or spend any time outside whatsoever, have a basic understanding that landscape diversity begets species diversity or biodiversity. And um, and then how that diversity transfers through trophic levels is something that I find quite interesting. So when we think about this positive correlation between habitat diversity and taxonomic diversity, it isn't always a straight line, of course, because nothing in ecology is ever a straight line. But that general pattern does hold wherever it has been studied. So it holds across biomes, it holds across spatial scales, whether it be your own backyard or the continent, and it holds across taxonomic groups. It's true for beetles, it's true for ants, it's true for birds, it's true for primates. And unsurprisingly, the targeting of habitat diversity by land trusts or other conservation organizations is also a relatively common phenomenon worldwide where conservation or land trusts operate. And most organizations, if they have the funds and or wherewithal to do so, target habitat diversity both within properties to try and maximize the biodiversity or conservation value of a single property, and also at a slightly larger scale, trying to maximize habitat diversity across the holdings of a given conservation organization or land trust. None of this discounts the goal of acquiring properties to target one habitat type or to target the habitat type of a single rare species or species of interest. That is also a laudable goal and one that has been shown to be successful in many, many cases. But as a general rule, and as we'll see with some of the data I'm going to present tonight, um, that broader targeting of habitat diversity appears to be a recipe success for conservation and land trust organizations. Just to pinpoint this again, Sarah showed one map. This is slightly closer version here. The image on the left is where the property is. And on the right, we have a zoom in from Google Earth that shows the approximate boundary of the property. One of the really nice things about Lot 16 is, and this is a real testament to the SILT board, is the positive relationship with the neighbor, who has in fact donated some land to make Lot 16 a bigger parcel, and has shown to have a real interest in Lot 16 being a successful haven for wildlife species, for many wildlife species. And the neighbor even, for those of you who came on the site tour, last summer, 2 summers ago, they all sort of blur in after a while. Graciously allowed to use their property as an access point to show folks. As Sarah mentioned, the initial slope here from the road up into this valley is quite steep. As a consequence, can be quite difficult to access for those limited mobility. And so when we were able to get to the site, Steiner let us drive our bus up there to get broader access. I want to talk today about two research programs that I've participated in with the blessing of SILT. After I talk about this first one, I will pause for some questions there, but again, feel free to put your hand. I can't actually see the chat, so I'm going to rely on Sarah to jump in if there are specific questions. But the first project I want to talk to you about is SILT's partnership or participation in what was known as the meta barcoding project, which is run by the Center of Biodiversity Genomics at the University of Guelph. Some of you may be familiar with barcoding as a rapid biodiversity assessment technique, rather than a direct focus on richness or species richness at the species, subspecies, or higher level orders of taxonomy. They're looking at genetic diversity as a proxy for species diversity. You'll see in here the phrase BIN, which is called a barcode index number, and that is thought to be a proxy for species diversity. And I've got the website here for the Biodiversity Genomics Center at Guelph. If you are that inclined, if you are a nerd with that inclination to get deep dive into molecular genetics and how that is really transforming a certain aspect of our biodiversity research worldwide, I encourage you to visit their site. There's some fascinating research projects, of which this is one. Their focus was in BC in 2023 with 16 sites across central and southern BC. They have sampled other geographic areas, mostly in central and eastern Canada over the last few years, but BC was the real focus. And the project involved setting up 2 malaise traps, which I will show you a picture of here in a second. And I set these up with the University of Guelph folks, and then I ran the traps for 5 months through May through September, and basically involved in getting to site at a minimum every 2 weeks, swapping out the sample jars, collecting the samples, and making sure that the chain of custody was secure so that the samples weren't contaminated in any way. So this is what a malaise trap is. It's a tent with a mesh bottom and a white fabric top. And the attractant of this system is the UV reflectance of the white portion of this. As many of you might know, most arthropods their visual spectra are highly weighted towards ultraviolet, and this really does turn out to show up as a vibrant beacon on the landscape that is active as a beacon both during daylight and dark hours. Most insects, when they encounter a barrier, tend to crawl upwards. And what you can see up here in the top, at the apex of the tent, is the sample jar which is in this case a jar full of ethanol with a fixative that prevents the DNA from degrading as part of the normal decomposition process. And so I would go to site every 2 weeks and swap out these sample jars. And I have to thank my wife for being willing to let me store these in the freezer for the duration of the summer. Although she did say that sealed jars of ethanol is better to have in the freezer than bats and caribou scat, which are often the things I have in the freezer. So we have we had two sampling stations for the purposes of this for the research. Both were in the riparian cottonwood forest that follows the ravine. They were relatively close to the bottom of the project area. And the reason for this was twofold, one of which was ease of access for setting up the traps and for swapping out the sample jars, and also trying to target areas with the maximal probability of capture. So the two sites were separated by about 500 meters in spatial, in horizontal distance, and about 50 meters in elevation. And what we see here are the primary results from that assessment over 2023. On the left-hand side, you see a Venn diagram. So BS1, which was the lower site, BS2, which is at the higher site. The numbers in the Venn diagram are the number of BINs, barcode index numbers. So there was the lower site had 1,208 BINs, and the upper site had 1,023. This is not directly comparable one-to-one to species richness, but it is pretty darn close. So what you can see immediately is that there is a super high arthropod diversity in the lower portions of Bourguiba Springs, with a conservative estimate of approximately 1,100 different species. I'll show you the breakdown of what groups of insects were found there. The chart on the right shows both the variation in the capture rates. Those are the bars. So the greeny blue bars are the lower site, and the yellowish bars are the upper site. And then the lines are the wet weight of the samples, which gives you an indication of how dense the sample was for that given day. It's a proxy for abundance in this particular case. And what you can see here is that, with the exception of one sampling period, the lower site typically had more diversity and more mass of arthropod biomass in it, but it did fluctuate across the year, with a noticeable dip in June of 2023. You're going to see this chart on the right again when I bring up the bat results from 2024. So again, take-home message here: a tremendous amount of diversity. The University of Guelph folks were not expecting this from where we were sampling. And the other point is how different the two sites were, given how how short the separation between the sites is. So even at that short spatial scale, shifting up in elevation, with the second site being a little bit more exposed than the first site, can have a pretty profound effect on diversity in this region. So who did we catch? Well, starting with the non-insect group, there were a number of arachnids, meaning that they found a way up into the trap, even though the trap didn't necessarily touch the ground at all times of year. Of these bins, there were 30 attributed to Araneae spiders, and several mites, Again, very persistent little things to get all the way up to the top of a malaise trap. And against all odds, Colambula, a springtail, which I'm guessing crawled on the back of a grasshopper and got up that way. But again, a really interesting um, testament to how powerful the attractiveness of that sort of UV reflective white part of the malaise trap can be. One of the spiders that was captured on site is this Sergolius columbianus, one of the ant mimics, an interesting spider. We also had some of the mesostigmata mites. This is Varroa destructor. This is a plant parasite, and then some Tribitiformes. We didn't actually catch this species, Trombidium holoceri, but these are the types of mites, and this is the group of mites that those of you who have had the misfortune to get chiggers, they belong in this group of mites. So a really interesting diversity, and of the 30 spiders, a really even diversity among crab spiders, jumping spiders, ground spiders, wolf spiders, etc. Not many orb weavers. There were only 2 in the orb weaver family here, but the spiders we did catch were the ones that tend to move around a lot. And as most of you can imagine, orb weavers tend to be pretty stationary throughout their life cycles. The next slide violates one of my cardinal rules, is to not have too many words on the slide, but it really does give a good indication of the diversity on site. Here's the insect list, right? And so we've got everything from beetles to earwigs through stoneflies and snakeflies. If you look at the numbers here in the parentheses, you'll see that, as expected, Diptera, true flies dominate the diversity, with Lepidoptera and Hymenoptera coming a distant second. This also shows despite the power of the UV reflectance of the white part of a malaise trap, it does show some bias towards winged insects in particular. Coleoptera is one of the most diverse groups, if not the most diverse groups of insects in North America, and are not highly represented in the sample. There were a few species at risk captured in this sampling. All of them were Lepidoptera, and all but Acaroides here were leaf miner moths of a variety of types. So a lot of moths that are so poorly understood that many of them don't have English names, but are recognized by the BC Conservation Data Center as blue listed. This is the Acari here. Acaroides is one of the subspecies is the Mission Blue, which is a species some of you may be familiar with, but one of the lovely leps that we do have in the area. I have confirmed several hair streaks, Bear's hair streak and others, half moon hair streak, on the property just through visual observations. They were not captured during the sampling. And just because I think they're great, the other picture I decided to show you was of a gula bicolor, one of the more common snake flies we have in the South Okanagan, which was the one of the 3 Rhiphidiopteras that were captured as part of this sampling. So it's an interesting sampling technique. It is an extractive sampling technique. These insects do not live to tell the tale of their adventure. But Al Pete and the board did reach out to many experts throughout the province when approached by the University of Guelph to talk about their concerns about the sampling, and were given the thumbs up by the folks that they reached out to. And what I think is impressive about this sample, and this is I'm early stages now with this analysis, but in the species here, there are species that have not been recorded in BC before, at least not in the provincial data set. There are some observations of some of these species in iNaturalist, but they've not been formally vetted through provincial systems. And so I'm going to be working through this long species list over the next little while, really highlighting those species that we have very poor understanding of their distribution in British Columbia, and highlighting Lot 16, Bourguiba Springs, as perhaps a hotspot for some of these species, if not the only known location in BC and or Canada for some of these species. So stay tuned there for that work. I'm going to pause there before going on to the second project, which was focused on things that eat arthropods.
That's amazing, Jason. I actually never knew that there was such an animal called a snake, a snake bug, um,
Snakefly,
the snake fly.
they're
Yeah,
awesome.
yeah, that's pretty amazing. And actually, the question that I had for you was about if there was some that had not been reported yet and what that kind of means. So yeah, so at this moment, I don't actually have any questions in the Q&A, but we will take a second because I know it takes a moment sometimes for people to come up with questions. It is a good opportunity, and Jason doesn't bite. You can definitely ask him anything, no holds bar.
Yeah,
Um, I think
yeah,
it's
we'll have time at the end too if
yeah,
folks can't do anything right now. And again, and I am in the process of, as I mentioned, drafting a report about all of this for SILT and to be shared at their discretion.
yeah. I mean, I suspect that you'll be hopefully publishing some of this work too.
Right.
And I imagine the biodiversity project is also very excited.
Yeah.
Okay, well, we won't belabor it, but
I do see a few things in the chat, not the Q&A
yeah, that's just people just chit-chatting.
chatting.
Okay,
Great.
there
Well,
is a
it's
question.
almost puck drop for the Oilers, so.
There is one question here from Lindsay, one of our board members. How many species do you estimate are new to BC?
Based on the quick scan, and the first scan I did was for species at risk, so comparing the species list that was provided to to the red and blue list from the CDC and the COSIDIC list. And then I did a quick scan of like, I've never heard of that before. And, looking it up on the CDC website of just beetles in BC. And so I would, at my quick glance, I would guess 40.
Wow. Yeah.
That now some of them might be just taxonomic name changes that have not been fully captured either one way or the other. Um, some of them might be the barcoding system is not perfect, right? It's as good as the test. It's as good as the training data, right? And so there is going to be some slush there, and that's why it's going to take a bit of time to sort of go through these one by one, reach out to the experts, um, those kinds of things. But there are also, in Dipter in particular, so 872 barcode index numbers. Half of those were just like midge, Ceratoponidae, right, or Chironomidae. And so that means one of two things. One of which is the sample was enough to demonstrate that it was a distinct sample, distinct sequence, but not distinct enough to match to a existing record of a sequence match, or there is no match
Hmm.
and it's new or has not been sampled before,
Wow.
which in the world of taxonomy is kind of the same thing.
Yeah.
Right. And so, and my sense from the University of Guelph folks is that's what's really got them jazzed up too, is just the sheer number of unknowns in the sample coming from And it wasn't just this site in BC. There were several other sites in BC that had a lot of unknowns and some of these hyper diverse groups, which means that there's if you thought taxonomy is dead, just study really tiny flies.
Yes, well, hopefully you can name one after SILT or maybe our founding.
Yeah, no, it's like
Members.
Ceratoponagus siltonii or something
Yeah.
like that, right? Yeah.
Okay, so I have another question here for
Okay.
you.
Yep.
Jason, do you have a picture of the trap up close? How do the insects enter the bottle and not escape and or eat each other or eat other insects?
Um, I don't have a close up of the top, but I can describe it. And so 100% insects are probably eating each other. Every time I went to sample there were usually 6 or 7 meadowhawks, one of our native dragonfly group, around the trap, just doing laps. So almost certainly they had figured out that there was a magnet here for things they like to eat. Um, they're at the very top of the Malaise trap. Here, I'll just go back quickly here. Yeah. At the very top of the Malaise trap is a an opening. So they fly in, they flutter around, they finally find their way to the inside on the white, and they crawl up because they crawl up, and there's only one way to go. And when they get to the top, they get to an opening, and if they keep going, they just fall into the jar. The estimate of the capture success of Malaise traps ranges from 30% to 70% of insects that hit the trap that end up in the bottle.
That's pretty good, actually.
So yeah, it's not bad. Still, so it's an underestimate of probably biomass more than it's an underestimate of species. But as you can see from this way this trap is set up, it's not really going to sample I shouldn't say that. It sampled a springtail. But there were no millipedes or centipedes, for
Yeah, they
example.
don't crawl up it, right? Like there's a
Well, some of them clearly do because they got spiders,
yeah.
right? So, and woe betide us if spiders ever figure out how to fly in in a directional sense. Maybe spiders dropped, right, with their silk, silk lines and the like. But yeah, it's a passive trap in that sense. You can
Okay.
sometimes some folks with Malaise traps, they'll put a pheromone. If they're targeting a particular species, they'll put a pheromone jar right there, and the insects climb up to the pheromone. But in a broadcast sample like this, they just let um, general behavior win.
Yeah. Okay, I have another question for you
Yep.
from Gord Wilson, one of our board members.
Mhm.
When you first looked at the site, would you have thought that there were that many different species of bugs and critters? There's a little happy face saying non-biologists speak on the property. So, was there as
Yeah,
much as you thought?
yeah. My first time there, I was like, "This is cool." Right. It's clearly a, there's a diverse bunch of habitats in a very small space. Kudos to the SILT team for getting this done. Because it's one of the I've not seen its like
Mm.
in on certainly not on the western slope, like this one is. And so I was expecting to be surprised. Um, this sort of exceeded that expectation by like 100%.
Wow, that's amazing.
This is about double what I was expecting
Okay,
based on the Malaise trapping I've done before.
that's amazing. Okay, so we've answered all the questions for now. So, I think we can move on to your second half. And then,
All
of course,
right,
if people still have questions to go back. We can always go back to any of this stuff too. So,
great.
thank you, Jason.
Okay,
And people are saying
let's
thank you for your responses.
no good. Let's fast forward to 2024. I had big plans to do map banding, bird counts, and all sorts of things. And then I gave my head a shake and said, let's just do one thing. And so I decided to do some bat monitoring. And Lindsay, one of the SILT board members, came up with me and kept me company while I set the detectors out and checked them. So this is again a map of the property. just so you're a reminder, I thought I'd put a map on with the locations. Maybe I did and forgot. Um, uh, Down here, so where the lower triangle here, where the lower bat detector is where the second of the malaise traps was the year before, and the first malaise trap was just down here a little bit lower. So this lower bat detector was put right in the riparian area, and then the upper bat detector I put in a sumac right underneath a major rockery outcrop right here. And the desire here was to try and catch as many different bat species who potentially could be using the project area as I could. I borrowed a couple bat detectors from EcoLogic, my company. It's a nice thing about being the owner of the company is you don't have to ask super permission. Two wildlife acoustic detectors, and I chose to implement them as full spectrum detectors. A little bit nerd speak, it gives a much better harmonic structure to the sonograms, the ultrasonic sonograms that it records, which allows for a better differentiation among call signatures of closely related species like many of the Myotis bats. Um, and the yeah, that's the easy answer. Um, so acoustic analysis is fairly well automated these days. Wildlife Acoustics has a proprietary software called Kaleidoscope, and the machine learning algorithms that are used to analyze these data are really quite good, and they do give a confidence interval for how confident the AI is that the species that they're tagging is this indeed that species. I have my own professional threshold where, in my experience, when the machine learning algorithm has a confidence over X, in my experience, they're generally bang on. Anything under X requires either visual inspection, looking at the individual sonograms, or lumping into different frequency classes. And I'll talk a little bit more about that. And so this is what these sonograms look like. And you see the frequency on the x-axis here and the amplitude or the on the or the frequency rather on the y-axis. And you get these little sweeps in here, right? These dots just refer to points along that sweep that the program is using to calculate certain parameters. And in this particular case, there is only one species whose calls terminate below 10 kilohertz, and that's this kid right here, spotted bat. So you do this for the number of calls, and I think over the summer of 2024, I think I recorded across the two detectors 30,000 bat calls, some of which turned out to be insects, which I was able to filter out. And but a good majority of them I was able to A, confirm were bats, and B, was able to generate a species list. And if you like bats and you like good return on your investment, SILT, for purchasing this property, this is the species list for the sampling in 2022. 1, 2, 3, 4, 5, 6, 9. What's that? 9, 12, 13, 14 different species, which is pretty much all the bats in BC, with the exception of Myotis centroptentrinalis, northern Myotis, and eastern red bat. So a really impressive diversity here, and there's a couple things highlighted here. So these are all the species for which I got at least one call that I was certain was a call of that species. As I mentioned, you're not certain about the species attribution of every call, but so then you end up lumping into low frequency bats, middle frequency bats, and high frequency bats. So the species diversity is just notable by itself. I don't know of any other site that has this species diversity in BC, period. The red species here are either, in the case of little brown myotis, not sure why I hyphenated that, a little typo there. That's endangered on the Species of Risk Act, the Federal Species of Risk Act. Pallid bat is on the provincial red list. Probably my favorite bat in the Okanagan. Silver-haired bat and hoary bat are both candidates for listing as endangered on the Species of Risk Act federally. The blue species are, surprise, blue listed species in BC, the Conservation Data Center. And the two underlying species here are species for whom in the last 20 years, we're seeing a dramatic northward range expansion. These species are now considered to be regular breeders in BC, whereas even as early as 10 years ago, you'd better have a bat in your hand if you were going to say that you had seen this species. But more and more folks are reporting these, and these call signatures that are in the algorithm of the Kaleidoscope program are widely considered to be valid. And so I wasn't surprised to see these species pop up on this list, but still was a pleasant occurrence nonetheless. Not all of these species are going to be breeding at SILT. In fact, I would imagine most of them are moving through, silver-haired hoary bat in particular. But the spotted bat, pallid bat, the myotis are almost certainly breeding in the area. For those of you who are bat nerds, this species name might be different for you. This is a split from what was Myotis ciliolabrum, the western small-footed bat, and they've split sort of the population west of the Rockies and east of the Rockies here. Not everybody agrees with this, but Corey Lawson, who I consider to be one of the top, and along with Jared Hobbs and others, consider this to be a valid taxon, and I'm not going to gainsay them for sure. Here is just a snapshot of the seasonality. So I have spring bat activity and fall bat activity, and the orange lines are the variation at the lower detector, which is smack in the riparian zone, and the blue is the variation up higher in the rocky outcrop area. And the patterns are as you would expect, right? More insects in the riparian areas, more water in the riparian areas leads to additional activity in these areas. And again, these are not the number of individuals, these are the number of calls that were detected, which is considered to be a proxy of number of individuals. And you see this real striking peak in June, in early June. This peak is this particular peak is due almost exclusively to the arrival or emergence of Myotis californicus, California myotis, by far and away the most common species detected here, although you see a similar peak. You see another peak here in the area. Fall bat activity, a little bit lower. This can mean a couple of things, one of which is those species that do migrate perhaps have a different northward versus southward path on their migration, or once breeding has ceased, like in many organisms, organisms tend to disperse away from their natal areas and they're not as tied to one location, which may increase things here. Sarah.
Two questions for you. The first question is my own, and I'm kind of just curious about, you mentioned earlier that most of these bats are just moving through.
Potentially, yeah.
Are there particular restoration actions that you think SILT could take to make it a more hospitable environment, or maybe for the local breeding species? Do you think that bat boxes would work, or is there other?
Yeah, not a lot of these species like bat boxes. There's a few different options we could take. Really, the best thing is just to let the cottonwoods age, right? And don't take them down as danger trees.
Yeah.
right? Which means you may need to close off that bottom access, right, once some of these cottonwoods go. But a lot of these species they'll be roosting in crevices and rocks. They'll be roosting in, behind the bark. And so it's already a pretty diverse spot. And the fact that, migration habitat for bats, as with birds, is super valuable,
Mm.
right? We hazard a guess that probably 80% of bat mortality happens during migration, much like 90% of songbird mortality happens during migration. And so these stopover points are absolutely critical. So I wouldn't stress too much about trying to increase breeding habitat as opposed to just maintaining a refuge for these species as they move through as we all know in the Okanagan, a highly modified and deeply pressured ecosystem.
So this kind of nicely dovetails to Gord's question. Would all of these bat species move between summer and winter habitats?
No, some migrate great distances. Silver-haired hoary bat migrate. They'll migrate. They'll breed just a tree line in the boreal forest, and they'll migrate to California, for example. Although we're seeing those migration paths shorten as global temperatures change. I had silver-haired bats try to overwinter here at my house in Oliver. Did not go well. Um, for them, unfortunately. Most of the myotis do smaller migrations and then hibernate. And we do see some species that hibernate in the Okanagan don't hibernate on the coast. There's enough, the temperatures don't get cold enough often enough for them to have to go fully into torpor. So a lot of these species could be hibernating locally, including in many of the rock crevices. If they go deep enough and to provide a stable microclimate, I wouldn't be surprised if many of these myotis species are either hibernating in the rocks or hibernating in local abandoned barns, and the like. A lot of these species will hibernate in human structures.
That's great. Okay, thank you. And Gord, if I think that answered your question, if not, just you can type another
Right.
expanded. Okay, thank you.
And then we could, admittedly, these are two different years, 2024 on the left and 2023 on the right. You can start to see some patterns between, activity patterns of bats and activity patterns or availability of prey. Granted, the x-axes are different here, so I've just added some boxes to sort of indicate concordance here between these two, right? Where you see, peak back activity with a good chunk of insects, a drop in bat activity as insect abundance drops as well. We do have another pickup in the summer. I wasn't monitoring over the summer, so I wasn't able to show, what that looked like. But there's a nice little snapshot of, as you would expect, bat activity is higher when insect activity is also higher. So where does that leave us, right? At 4 minutes to 8 o'clock. I think, there's three things three ecological first principles that I think were nicely verified by both studies that SILT has supported on this property. One, even small scale differences in habitat heterogeneity can increase taxonomic diversity, as we've seen for the for me, quite astounding lack of overlap between the insect samples at the two malaise traps. Small to mid-sized properties are 100% worthy of protection if thoughtfully selected based on location, contribution to the local network of habitat diversity, and landscape rarity. And finally, the use of umbrella species, in this case, bighorn sheep, that was one of the real motivators behind this property acquisition, or species groups like bats, can really have substantial down gradient positive effects on the species whose home ranges or behaviors exist at smaller spatial and temporal scales, get sort of swept along by the positive benefits of focusing on some of these species with larger footprints. And so the punchline is, good job, SILT. Right? This is a really great example of how a concerted effort amongst like-minded individuals, like-minded organizations who care deeply about the environment and as deeply about using the donations and grant dollars that have come their way to best effect. And so for those of you who, are donors or thinking about donors, this property shows that the SILT team definitely has the right idea, especially here in the South Okanagan. And for those of you who are interested in, braving the poison ivy patches at the bottom of the property, please stay tuned for whatever I convince SILT to let me do next year. Thanks very much for your attention, folks, and I'm happy to take a few questions here as we end.